Okay, we are going to do this question number 7 now. So question number 7, it's about the brain and the neurotransmitters. Serotonin is produced by neurons in the brain. How many of the following statement about serotonin are correct? Number 1, L-DOPA can be converted into serotonin in the brain. This is wrong because we know that L-DOPA is the treatment for Parkinson's disease because L-DOPA can cross the blood-brain barrier, and after that, L-DOPA can convert into dopamine. The dopamine level increase, and this helps Parkinson's disease patients because they have a problem of dopamine. Dopamine goes down, and they develop this muscle tremors and rigidity. Right, next one, serotonin is a neurotransmitter. Of course, yes. Serotonin work like this. This is a pre-synaptic neuron. This is a post-synaptic neurons. Upon stimulation of the action potentials, calcium channel open and calcium bind with the vesicles, and vesicle reach to the presynaptic membrane, and serotonin is released. So this is wrong because it is produced by presynaptic neurons. So answer is B. There are 14 different serotonin receptors found in human nervous system. 3 different genes are produced to produce these receptors. Explain how 3 genes can produce 14 different receptors for serotonin. I think there's a small problem with this question with most of the students, because this is unit 2 question. Now, if you have done the AS, most of the time you forget this one because this is a little bit of an advanced theory, and you can't remember this. So we have learned in unit 2, if there is a gene, this gene has 2 regions. One is the red color one, they are called exons, and blue color one, introns. So I write here, this is exon, and this is introns. Okay, then this gene transcribe to a pre-RNA, pre-messenger RNA. Okay, the pre-messenger RNA, as you know, it's again a single strand, and it could be like this again, drawing from here to here, and it copies everything again. So here we have exon, here we have this exon, here we have this exon. Okay, after that, there's something called post-transcriptional modification, right? Post-transcriptional modification, PTM. Right. What's the first thing in post-transcriptional modification? There's a group of enzymes known as spliceosome. They can bind to these regions. You can see these regions between introns and exons, and these are the enzyme complex. After that, they're going to hydrolyze the phosphodiester bond. As a result, they're going to cleave these exons and intron into pieces. Here one, and here one. This one, this, and now this. So they chopped down these exons and introns. After that, resulted in exons separated and introns separated. Now, when we look this uh, exon. So exon, let's say this exon is A, B, and C. A, B, and C. A, B, and C. Now there is a possibility these exons can reunion in many different ways. For example, A, B, C, O, C, O, B, C, A, O, C, A, B, O, A, B, O, B, C. So they have a lot of opportunities now. They can reunion in many different ways. So A, B, C is the most appropriate now according to this order, or could be a C, A, B, or B, A, C, A, B, A, C. Now, when it's done, this is called mature mRNA. So mature mRNA leaving the nucleus to the cytoplasm, and from the cytoplasm, they can produce a polypeptide chain with different sequence, different number polypeptide chain. For example, these two number is same, but the sequence is different. And if you take this one, number is different, polypeptide chain amino acid number is different than this one. So then different number, different order of polypeptide chain produced, and these polypeptide chain produced to get the tertiary structure, and they become receptors for serotonin. And then different proteins can produce, different receptors can produce. With 3 genes, 14 different receptors can be produced. Okay, next question. Concentration of serotonin. Okay, we read the question. The effect of ecstasy MDMA on serotonin concentration in rats was investigated. Rats were given concentration of MDMA from mouth, and then concentration increase in this brain and spinal cord fluid called cerebrospinal fluid, and analyzed 24 hours after the last dose of MDMA. The graph shows the concentration. Okay, concentration line of best fit and result increase, decrease, increase, and we have best fit curve. Determine the effect MDA on the serotonin level in the cerebrospinal fluid. Okay, determine the effect of MDA. MDMA concentration increases, concentration of serotonin also increases as a positive correlation. So for the second mark, we can calculate the percentage increase. For example, starting point is 250 here, and then this one is a 260, 70, 80, 90. Okay, 250. 1, 2, 3, 4, 5. So 250 and 300 here. So this could be 260. And this could be 390. So we can calculate percentage increase. 390 minus 260 is a different, and divided by initial value to get the percentage increase value. So this could be 550 divided by 260 into 100. So I use a calculator. So if I use a calculator, 150 divided by 260 multiplied by 100, it's about 58%. 58% increase. Okay, determine the effect of MD on the serotonin level in the cerebros. Okay, so now next question. Predict the concentration of serotonin for dose 17.5 milligram per kilogram of MDMA. Right. I think the best fit curve is given here. So we had to extrapolate best fit curve until this point, 17.5, and we get the concentration in 400 milligram for DMQ. Sorry, CMQ. We need to convert this 400 milligram into a gram here. To do that, we have to divide by 1000. It's a 0.4 gram per CMQ. Okay. Right, we are coming to the last question now. Explain how MDA taken by mouth could affect the level of serotonin in the brain. Use the information in the question to support your answer. Now, when we take this kind of a question, these are not difficult questions. So when we take a drug is absorbed into the blood and then goes to the brain, and then it makes the effect. So now most of the student, they cannot get this simple idea. They come up with many different answers. So either MDMA, we have no idea. Usually, many things are digested, or small chemicals they can absorb. So they are absorbed into blood, then transport to brain and penetrate blood-brain barrier of the brain, and then bind to the postsynaptic neuron. So it's like this. So now this is a presynaptic and there's a postsynaptic. That's a receptor. Now MDMA can bind here, and then we can make a new action potential and make the effect, because the action potential can trigger the neurons of the certain places of the brain, and they can make an effect. Okay, done this question. So we will continue other questions.